Now, I'd like to introduce Kimball Garrett, who will introduce our guest for tonight. Kimball?
Okay, thank you, Ron. I hope you can hear me over my internet connection here. But anyway, I am delighted to introduce Allison Schultz, our speaker tonight. Allison joined the staff of the Natural History Museum in September of 2018 as Assistant Curator of Birds. She follows a long line of bird curators there, some names you might recognize from Luther Wyman and George Willett to Ken Steger, Ralph Schreiber, and Ken Campbell. Allison got her undergraduate degree at the University of California, Berkeley, where taking a natural history of vertebrates course really spurred her interest in birds and other aspects of natural history. She did an undergraduate thesis that was actually published in the Condor, which was a look at the avifauna of the UC Berkeley campus, comparing older notes from the Grinnell era and some intermediate times with recent bird surveys. So it's really was an important part of that Grinnell resurvey effort that MVZ had done. She went on to San Diego State University for her master's, where she worked with Kevin Burns. Kevin had a group of researchers there who were responsible in many ways for an upheaval in taxonomy, which in retrospect made perfect sense, which of course is that the tanagers that we have are in fact not tanagers, but they're in the cardinal family. But Kevin also spearheaded a lot of research on the true tanagers of the neotropics, and Allison did her master's thesis looking at the evolution of color patterns in a particular group of tanagers, and that really spurred her interest in subjects like avian coloration, avian vision, because it doesn't make any sense to study bird color unless you know how birds themselves are perceiving it. And a lot of really began some of that genomic work too. And then she went on to Harvard for her PhD under Scott Edwards. Again, really getting into genomics, and especially as part of her interest in kind of rapid evolutionary change in populations, looked at the response of house finches to emerging diseases in various parts of their range where they were exposed at different times. And that really launched her genomic work and her love of house finches, which, looking at her background, you probably have already guessed about already. So, and then she went on after her PhD, working a little longer at Harvard on some bioinformatics stuff. She's an absolute computer whiz. She knows everything about, she knows a lot about everything I don't know anything about. So it's great to have her at the Natural History Museum. Anyway, we're really delighted. Now at the museum, she's also very much involved in our Urban Nature Research Center, and looking at the nexus of community science and looking at urban birds and evolution in urban birds and so on, with a particular interest, and eventually applying her genomics background on looking at some of our naturalized non-native bird species. But tonight she'll be talking about her work for her PhD on house finches and their evolutionary response to disease. So again, we're all delighted that Allison is speaking to us tonight, and we look forward to her talk. So Allison, welcome.
Thank you so much for that introduction, Kimball. It's really a pleasure to be here to talk to you all, and I'm going to share my screen now. All right, so I'm going to be talking to you today about the role of infectious disease in shaping avian evolution. And so some of that will be about house finches, but some of it will be about birds in general. So as an evolutionary biologist, my goal is to really understand the processes that generate the incredible patterns of biodiversity that we observe across the avian tree of life. So there's a few ways that you can do this. One approach that commonly is taken is to study a trait of interest across a wide variety of species. So for example, here I'm indicating here's what we call a phylogeny or family tree, and we might see what patterns we can observe by looking at how these traits are correlated with geography or with abiotic factors like climate or biotic factors. And these correlations can generate hypotheses of what might underlie trait evolution, but by themselves, they can't really be used to infer causation. So another approach to take is to study evolution in real time. And this is often done using experimental evolution. So for example, I might take yeast or something like that and grow them up in the lab and look at how things are changing over time. However, that's hard to do in natural populations. And so in my research, I try and take both of these approaches and actually connect them by additional timescales, looking not just among species, but among populations of species or within different populations of species. And this arm of my research is really focused on a particular type of selective pressure, and that's pathogens. So virtually every organism is in a constant arms race with a variety of different pathogens, whether they be bacterial, viral, fungal, or eukaryotic in origin. So here we've got our little bird friend and our little virus friend, just to show you what that arms race means. Well, that means that the pathogen is going to be constantly evolving to be better at infecting a particular host or to jump to new hosts. This is something that we know intimately given the current situation. But it's not just a one-way street. So hosts are going to constantly be evolving to counter this, to be able to resist the hosts or to tolerate them, to resist the pathogen or to tolerate them. And so over time, this presents an arms race, just very much like building up weapons and becoming more and more extreme. And so one way how do we study this in real life? Well, one approach is to look at an organism's physiology or do experimental infections. It can be really difficult to know what's going on inside of an organism without those types of approaches. But another type of approach you can take is actually use genetic tools to infer this history of host-pathogen interactions. So we can use the genetics of an organism and look at what are the fingerprints of this interactions that we might be able to see. Over long time scales, we can use an approach called Comparative Genomics, and that's when we take genomes of different species. A genome is the entire genetic blueprint of an organism or all of the DNA. And we can infer, for example, how these organisms are related to each other in a family tree. And then we can look at how the actual genetic, the genes actually might be evolving on this family tree. And we can also take organisms where we can take in a single population or a single species, we can take the genomes of different individuals and infer really recent signatures of pathogen-mediated selection. So you can use comparative genomics to do long-term studies or what we call population genomics to do short-term studies. We know from studies in mammals and humans especially, humans are arguably the best studied organism from a population genetic perspective. By comparing genomes from humans in different populations, researchers found that pathogens are actually one of the strongest predictors of human genetic variation. And so this means that pathogens are actually one of the strongest factors shaping the human genome. Other studies have found evidence that evolution due to pathogens that cause diseases like malaria, tuberculosis, smallpox, leprosy, diphtheria, cholera, and AIDS can all be found in human genomes. So we can see evidence of human evolution due to a variety of different pathogens. But to date, many of these studies have focused on organisms with direct medical implications like humans, broader mammals, fruit flies, and not a lot have studied non-model organisms, and that's where my research really comes into play. So I'm going to be looking at evolution, host pathogen evolution over 2 timescales. So first, I'm going to take this long timescale view using comparative genomics, and I'm going to ask what are the genomic consequences of pathogens over long timescales. And when I say long, I mean evolutionarily long. We're talking about, across birds as a whole or across many millions of years. And so more specifically, I want to ask this question, which is, do pathogens target the same genes? And what do I mean by that? Well, let's take an example that might be a little bit familiar to us given the news these days, and that's COVID-19 and this protein called ACE2. So you might have heard of ACE2. This is just a little graphic of how COVID can enter a cell. Well, ACE2 is this receptor. There are 2 different ways COVID can enter. And you might have heard that of the spike protein on COVID. And what happens is that ACE2 is going to bind to the coronavirus spike protein, and that's going to allow it to enter the cell and then to replicate. And so essentially what this happened, this ACE2, it's not there just to let COVID into the cells. Of course, it has important functions in our in our immune system and our physiologic responses. But COVID uses this as a key to get in. So over time, you could imagine, if we were evolving with COVID or with another coronavirus that can use ACE2, we would evolve so this would no longer recognize the COVID, but COVID might then evolve compensatory mutations that's going to allow it to maybe bind tighter. We can actually see this happening in real time with these mutations that you might have heard of. There's the UK variant, this B117 mutations. That's actually mutations happening on the spike protein that might allow it to bind closer with our ACE2 receptor. And so this is just one example, but you can imagine there are many different pathogens that might be trying to subvert our immune system or an organism's immune system in many different ways. And over long evolutionary timescales, we see what's called positive selection, or we see more functional changes in these genes that have these relationships than you would expect just by chance. So let's go to this cartoon. This is a pathway. These are what we would think of as receptor cells. So these might be recognizing pathogens. Genes interact in networks. And so this receptors, they're going to pass a signal to different genes, which are going to kind of have this cascade of signals, which are eventually going to go to what we call effector genes, which are then going to attack that pathogen and try and get it out of your cells. Okay, so that's a lot of background. So do pathogens target the same genes? What I mean by that is over long timescales, there are two possibilities. One is that host pathogen hotspots are shared. So it's possible that the same pathogens might target the same genes and use those specific, maybe they're weak points in our immune system to try and get in, try and subvert our immune system. And so that would mean we would expect to see signatures of selection at those genes over long evolutionary timescales. However, it's also possible that host pathogen hotspots are species specific. That might mean different pathogens target different genes. Maybe there's different kind of weak points, and this might occur differently in different species. So that would mean that if we're looking on a very recent timescale basis, so let's say in one population, so maybe in a population that's just has a new pathogen, for example, then we might see selection in a particular gene, but over long evolutionary timescales, so over millions of years, we wouldn't necessarily see these same signatures of selection. Okay, I know this is a bird talk, but I've talked a lot about disease in general, but birds are actually really excellent models for studies of disease ecology and immune system evolution. Here I've got just a map depicting some migratory pathways of birds. And so how does this relate to disease? Well, birds are moving around. They're moving around constantly every year. And that means that we're going to have different species mixing with each other. Very similar to how humans, we travel the globe, and that allowed diseases to spread much more quickly than if we were just staying in one place all the time. So birds travel. Second is we actually know quite a lot about the bird immune system because birds are important in the poultry industry and because of the possibility of bird diseases jumping into humans. So people have done a lot of medical research. And as a researcher interested in just the biology of these things, I could take advantage of those previous studies that have really categorized the bird immune system. And so what did my colleagues and we analyzed. What we did was, okay, we took 39 species from across the bird tree of life. This is what we call, again, that phylogeny, how those species are related. If you are just scanning, you can see it's a pretty nice selection of birds that address most of the different clades of birds that are out there. And we actually looked at 11,000 genes, and we scanned them for signatures of natural selection in birds. So we were looking for genes that had evidence of more functional changes than you would expect, which would be a signature of pathogen-mediated selection or just consistent signatures of selection through time across birds. Now, as I mentioned, genes often function in interacting networks. So one way that we made sense of this was to look at, how genes are connected together because these different networks have different functions. What do I mean by that? Well, we might have networks that are generally have functions with the immune system. There are networks that deal with metabolism, so the ability for birds to kind of break down different proteins or different carbohydrates to use them for energy. There are other functions, cellular functions like DNA repair. And so what we found is that of those 11,000 genes that we studied, about 10% of them showed strong evidence of positive selection. That was quite a lot, but what are these genes doing? That's why we needed these functional networks to actually say, do we actually have, particular functional networks that have more genes under selection than expected? What do I mean by that? Well, let's say the pink circles here are now genes under selection compared to the black circles, which are not under selection. Are they concentrated in any sort of functional area? So in this case, I would say this immune system pathway, that's got a lot more genes than I would expect, as if those were just kind of randomly distributed among the different pathways. So what do we see in birds? Well, I know it's a complicated looking network, but let me walk you through it. So each of these big circles is a particular network, and each of the small circles is a gene that's connected to that network. And you can see some networks share the same genes. What I want you to pay attention to is that there are a few different clumps. There's this blue clump here, there's this pink clump here, there's this green, small green, and this little yellow one. And so these generally have different functions. And what was really exciting when we were looking at this with this eye of host pathogen coevolution is that this biggest clump we see is actually immune. These are all immune system networks. So these are all different genes and different pathways that are involved in either fighting off a disease or signaling a disease, and different types of pathogens. And this really supports this idea that pathogens are targeting the same genes across species and probably across different types of pathogens as well. So this supports this that first possibility that we said, that there are shared hotspots among species that pathogens are targeting. Now, just as a small aside, this other little small clump, this pink clump up here, was pretty exciting to us. So this is involved in DNA replication and repair. Now, why is that cool? Well, one thing about birds that you might not know is that bird genomes are small. So as humans, our genomes are about 3 billion base pairs large. So about 3 billion nucleotides, A, T, C's, and G's. But birds actually only have about 1.2, between 1 and 1.2 billion. So bird genomes are about a third the size of mammal genomes. And within birds, there's variation in genome size, with hummingbirds having the smallest genomes and flightless birds like Polygenics having the largest genomes. Polygenics are things like emus and ostriches. And there's a hypothesis that the reason that birds have such small genomes is because of their ability to fly. And you might think, why does that happen? Is it because, DNA is lighter if you only have a third of the bases? Well, that's probably not the case. Probably that doesn't add up to really have much of a weight savings in birds. But one thing about flight is it's very metabolically costly. You have to be able to really gather your energy stores, because flight takes so much energy to do it well. And so think about within birds, what birds have the biggest metabolic, they use the most energy? That's hummingbirds. So this is also supported within hummingbirds. And so how does that relate to genome size? Well, if you need to get energy really quickly, you're going to need to access these different genes in your genome really quickly. So you want a lot less of the kind of extra stuff. So a lot less of, in our genome, there are a bunch of repeat elements that don't really do anything. And so birds have a lot less of that. Their genomes are simply more streamlined. And now how does that tie back into host pathogen coevolution? Well, there's a hypothesis that homologous recombination, that's how when we get between our two copies of our chromosomes, they'll exchange genetic information that keeps kind of, that's a topic that's more complicated that I want to go into right now. But that's been hypothesized to be almost like an immune response to keeping bird genomes small. So it was really exciting to find evidence for selection in these genes. So it's not just that they have these relationships with external pathogens, but also internal pathogens, which are these segments of DNA that jump and copy themselves that we're not as good at getting rid of in our genome as birds are. Okay. So moving on. So back to pathogens like influenza A, West Nile virus, E. coli, Plasmodium. So, looking at these pathways is one way to do it, but another way to kind of see what are the genes doing that are under selection is actually to take data where an individual has been infected with a disease and you see which genes are activated. So that's a technique called transcriptomics. And so here we took data from previous data sets. So we didn't do any of the infections ourselves, but we kind of gathered data from across a number of species and a number of different pathogens. And we had 3 different categories for each pathogen. We had genes that were upregulated. So that means if you're infected, these genes are activated. Genes that are downregulated, that means if you're infected, you actually have fewer copies of these genes. And genes where just it doesn't matter. You have the same number of genes active if you're infected or not. And then we looked at how many of genes in these different classes were in that pathogen data set. So if our hypothesis that genes important in host pathogen coevolution are more likely to be under selection, so if pathogens are targeting the same genes, we would expect a greater proportion in this upregulated category compared to down or none. And in fact, that's what we do see across all of these different pathogens, especially the viruses. You might say, what about these big blocks over here? But there were only 2 genes in this category and only 4 in this. So it was really that there just were very few down-regulated genes. So that's very exciting. So this is basically confirming our results that pathogens seem to be targeting the same genes across birds over long evolutionary timescales. So that's just what I said. Immune genes are enriched for signatures of positive selection, and this is also true in genes upregulated by pathogens. And so before I go forward with more recent timescales, I'm going to take a step back. One thing that we're interested in also is what happens over longer evolutionary timescales, say across birds and mammals. So a similar study to ours had come out in mammals that showed that 30% of genes that interact with viruses are more likely to be under adaptation. And this is also true for proteins or genes that interacted with parasites. They also showed different kinds of signatures. But one thing we wondered was, are the pathogens still interacting with the same genes in birds and in mammals? And so that has implications for the ability for a pathogen to, say, jump from a bird to a mammal or from a mammal to a bird. So we combined our data set, all the genes that we had sequenced. We found there were a number of, I think there were about 4,000 that were also sequenced in this data set. And the first thing we asked was just, was there any overlap in genes under selection in birds and mammals? So this is not taking into account those that are involved in host pathogen coevolution. Just, we have these genes under selection, so that means that there's probably some sort of functional implications. Do we see the same genes? Are they tending to be under selection in mammals as well? Or is something completely different happening in birds and mammals? And so in this graph, don't worry too much about this. This is just kind of statistical, making sure our results are robust. But any values above the line are going to mean that there's more overlap than it's expected. Below the line is going to be less overlap than expected. So what do we see? Well, we see kind of this is the most stringent cutoff, and as we relax that, the results hold true that there are more genes under selection in birds and mammals than you would expect just by chance. And so this is very cool. This means that some of the same processes that are shaping a bird genome are also shaping mammal genomes. Now the question is, what are those processes? Is it pathogens? Do we see kind of the same pathogens attacking the same genes or different genes? Well, I'm coming back to this same network diagram that I showed earlier. And what we did was then we asked, are any of these networks that we had, do they have more genes than you would expect under selection in birds and mammals than birds alone? And 2 actually popped up that did. They were influenza A and herpes simplex infection. So these are 2 viruses. So that this is really exciting. This means that not only do birds and mammals share some of the same genes under selection, but these are actually genes that are known to interact with viruses. And influenza A, that's bird flu. So that means that that these viruses are interacting with bird immune systems and mammal immune systems in very similar ways. So we can add that to our conclusion. And just to put into context, this is a phylogeny. I didn't create this. Sorry, I meant to put the citation down there and I forgot. But this is just showing these are different families of viruses. This is the host family tree on the left and the virus family tree on the right. Blue lines are viruses that originate in mammals. Green lines are lines that originate in birds. And the more crossing, that means the more jumping from one type of, from one clade to another clade. So something like these types of viruses, you can see mammals stay pretty much together, birds stay pretty much together. But something like this family of viruses, which is the one that contains influenza A, so like bird flu, you can see there are quite a lot of jumping between birds and mammals. And so this is just another way to kind of support this importance that we actually study, take into account both types of immune systems. Just for fun, here's Coronaviridae, and you can see birds really form their own clade and mammals form their own clade. There's really no overlap between bird and mammal coronaviruses, so there's not much of a worry of us giving birds coronaviruses or vice versa. Okay, I've talked to you guys a lot about genomics. I'm now going to go into a much more recent time series and bring it back to my friend, the house finch. So let's ask this question: what are the consequences of a novel pathogen on a host genome? Well, first of all, what we're really asking, how can a population evolve resistance to a pathogen? So here, this is my population of birds. Let's say they have a new pathogen that jumped to them. And let's say only the yellow birds were resistant to that pathogen. Well, over time, you would expect this population essentially to change to all yellow birds because of this pressure from this pathogen. So how does that happen? Well, there are a few different ways. One is called a hard sweep. So this is a selective sweep. And what this means, so these are genetic sequences in 5 different individuals. You notice that these are all blue and turquoise birds. None of them are yellow. But in a population, let's say a resistant one evolves and it's got a C here instead of a G. Well, in that new population that's all yellow birds, you would expect all of these bases now to be a C, because this is what's actually allowing them to be resistant to that new pathogen. So, what do I look for when I'm actually looking through the genome? Well, if I was to compare, what we call allele frequencies, that's the frequency of a base in the population. So in this, the allele frequency of A would be 2/5. Here in G, this would be 5/5. So this is just a way to look at, how many of these bases are in the population. If I was to look at the difference through time, I might expect there to be very little differences except at this C locus, because that is going to be completely different between these two populations. So I would see a very big spike there. Another possibility is what we call a soft sweep, and it's very similar to a hard sweep, except instead of a new mutation evolving, this mutation might already exist in the population. So there are going to be some individuals that are kind of pre-adapted to be resistant to this new disease. And so here for example, we already have 2 Cs in the population. And over time, this would become 5 Cs. So if we were to look at this difference before and after, we'd see, again, a spike, but it would be much smaller. So it would be just not as distinct as that hard sweep. The third possibility is what we call polygenic selection. Now, what this means is that instead of a sweep at one place, so maybe it's not just one gene that's allowing you to survive this disease, maybe it's a whole bunch of different genes. And so if I was, again, to look at differences before and after, I would see, more than one spike, and they would be much shorter. This is much harder to find in the data. And finally, the last possibility that I'm going to talk to you about is immigration. And so this, what does this mean? Well, this means that, let's say you have a population and you actually have that infection, but you also have individuals coming from outside that are resistant to that infection. So, when you could do before and after, it's not that we have just one place in the gene that's going to be different, but it's going to be all the places where these different where this yellow population, wherever they came from, and this original kind of blue and turquoise population. So we're going to see a lot more differentiation throughout the entire genome, not just at one or two or a few spots. Okay, so those are the possibilities. Now, how do we know which one is most likely? Well, we can look at the house finches. So house finches are really a model for studies of ecology and evolution. And why is that? Well, in 1994, a poultry-associated Microplasma gallisepticum jumped from poultry to house finches. Now I'm showing a chicken here. If we actually look at the genetic tree of the Microplasmas themselves, it's actually closest to turkey. So people think it's a turkey strain, but poultry in general. Now you might be familiar with this. This is the house finch conjunctivitis. And so it makes house finches have very red, weepy eyes. Their eyes get all swollen. And it's actually not this Mycoplasma. So I'm going to call it MG from now on, Microplasma gallisepticum. Conjunctivitis, all those terms mean the same thing. So it's not actually that the MG is killing the house finches. It's that if you're a little bird and your eyes swell shut and you can't see anything, your odds are getting away from a predator are not very high, right? So within 2 to 3 years, this MG spread throughout the entire eastern population of house finches, and there were up to 60% declines in some areas. And numbers really have stayed down by then, even though that first decline was very rapid. It took about 10 years for it to get to the western population, where it was much slower to spread, but it had a lower prevalence, but it's really sped up. I just look at, for example, iNaturalist, there are a number of different observations of Mycoplasma even this last year. So we have this situation where you have, very rapid spread and extreme declines and much slower spread and declines, but they've been a little bit more variable in the western population. And so I'm not the first person, of course, to study this system. This was, my PhD advisor, Scott Edwards. He and his collaborators, as well as some other groups, have really studied this system for a long time. The reason that we actually know so much about how this spread was through community science, through Project Feeder Watch, because conjunctivitis is such a visible disease. You can easily see if a house finch is sick or not. Community scientists really could track how this disease was spreading and exactly what it was doing to the population in a very nuanced way, which was actually very exciting. So, okay, so we have this disease that's been in the eastern population. The question was, have house finches evolved resistance to Mycoplasma at all? They were lost 60% of the population, so you might expect natural selection might have happened. Well, colleagues from Scott Edwards and Jeff Hill and some others, they took individuals from Alabama that had been with that population had been living with MG for about 10 years, and individuals from Arizona. This is before they ever saw MG. They had, this was before it got there. They infected them both with MG, and then measured the response. So how sick did the birds get? And so what they found was, both for resistance, that these birds from Arizona had less resistance. That means that they had more MG in their system. So MG was able to spread and develop much better in these birds that had never seen it before than these birds from Alabama that had been living with it for about 10 years. And similarly for tolerance. So tolerance means that for a given amount of MG, the symptoms were much greater in Arizona. So their eyes would be much more swollen if they had the same amount of MG in Arizona as in Alabama, where their symptoms were much smaller. So this suggests that house finches that had been evolving with MG for 10 years had evolved both resistance and tolerance to this disease. But the unanswered question is, how did they do that? Was it through one mutation, hard sweep? Maybe it already existed in the population. It was a soft sweep. Maybe it was multiple places in the genome, polygenic selection. Maybe it was immigration. Maybe, there was one population that was just resistant that spread throughout the entire East Coast, for example. So how can we measure a population from before something happened and after something happened? You need a time machine in order to do that. And luckily, we have one, which are museum collections. So museum collections are a window into the past. Every specimen that was collected at a particular place at a particular time is basically a little time machine to what that population was like at that point in time. And so we could use museum specimens of house finches that were collected in 1990 and 1991. Luckily for us, a colleague at the Royal Ontario Museum had just gone around and was planning on doing a house finch phylogeography study and had done really good sampling. And this was just 3 years before MG hit. So we had really good before data, as well as, then we could go out and collect after data. So in our experimental setup, we had this Western population, which again remembers the spread was much slower here before and after, and also the Eastern population, which has been exposed for longer and had a much sharper declines. So just to give you some specifics as to what our sampling was like, so we sampled 16 house finches per population in the before, 4 populations in the east, 2 populations in the west, and then 4 populations in the east and 2 in the west from 10 years after. And then enough time had gone by in the eastern population that we could also sample 20 years after. So we could see, not just one time point, but actually 2. So what's it like 10 generations after Microplasma, and what's it like 20 generations when a little bit more time has passed? And our ability to look at this is only going to increase through time. So how did house finches evolve resistance and tolerance to MG? Well, first thing we did was to look at differentiation through time. So here's that genome position on the x-axis, and then on the y-axis, any points that are above this line we're calling to be significant. So what we're looking for is, a big spike of points somewhere in the genome, or maybe a smaller spike of points. What do we find? We found nothing, no spike of points. So this was very sad. When you're a graduate student and you're plotting your data for the first time, it can be pretty disappointing when you don't see anything. And this was true both in the Eastern and the Western populations. But, what does that mean? All that means is that we can cross a couple of possibilities off of our list. We know it's not a hard sweep. We didn't see that big spike. We know it's not a soft sweep. We didn't see a smaller spike. But now the question is, was it polygenic selection or was it immigration? Well, before we can answer that, we have to go into a little bit more. If we want to learn about, immigration, what is the actual demographic history of the house finch? Because that plays into the story as well. So, out here in California, house finches are very common, and we're actually in the native range of house finches. So in 1870, house finches were introduced to Hawaii. They spread very quickly there. And in 1940 was when they got introduced to the East Coast. After that, they spread very rapidly in both areas to where this is the range map in approximately 1990. These days, these two ranges are very much closer together, and we're actually seeing some crossing over. So it'll be very exciting to kind of see how that plays out in terms of house finch genetics, exciting in the house finch world. But what are the consequences of these introduction events? Well, let's talk a little bit about from a genetic perspective. So when a population is introduced to a new area, they can undergo what's called a bottleneck. And what does that mean? Well, we're not going to be sampling all of the individuals and possibly only a small number of individuals. So it's possible that we might not capture all of the genetic variation when these house finches are moved from one place to another place. And in genetic terms, that means they have less genetic diversity and have undergone what's called a bottleneck. Another possibility is when birds or any organism moves to, if their population expands, they're actually much more likely to have mutations. And so here we can see a few new colors of birds introduced. And so we actually might see greater genetic diversity in some sense after an expansion compared to a bottleneck. Now, remember back to the East Coast house finches, well, they underwent a bottleneck followed by a big expansion. How did that affect their genetics? So in addition to this before and after question, we now have to add this question of differences between Western and Eastern populations. So in addition to this overall question, how did house finches resolve resistance and tolerance to MG? We want to ask, how is the demographic history of the house finch affected genetic variation? Specifically, I want to look at how are these populations structured through time and space? So do we see evidence of populations clustering together differently through time? That would suggest immigration probably had happened. And how did the bottlenecks and expansions due to MG or the introduction affect genetic diversity? So I used a technique called principal components analysis. So this, I've got, when I look across the genome, I've got about 30 million sites that are variable. That's a lot of sites. How do I collapse all of that information into a single graph? Well, this technique called PCA allows us to do that. So each point on this graph is going to be an individual, and the distance among points is going to just generally show how these individuals are related to each other. So I also had sequenced some cassins and purple finches, house finch relatives. So the first thing we did is look at how house finches, cassins finches, and purple finches separated. And you can see here's cassins finches, here's purple finches, here's house finches. They all separate by species. That's a good thing. That means that all of our house finches are house finches. Funny story, in my original data set, one of my house finches actually grouped over here with purple finches, and I went back to that specimen, and it was indeed a purple finch. So that was, a case where sometimes your samples might be misclassified, and it's really valuable to have those museum voucher specimens so you can go check that. But let's look specifically at house finches now. Well, what about when we look at genetic variation just within house finches? Well, the first split is between Eastern and Western population. So that makes sense because you would expect that Eastern individuals, because of that bottleneck, they'd be all more similar to each other than the Western individuals. And within this, they basically are differentiated by location, by geography versus time. Now, a really cool thing, if we look at kind of the averages from each of these different locations that we sampled in different time periods, you can see that it actually forms a linear relationship from the distance where they were introduced, which was Long Island, New York, which means that as house finches, as the population expanded further and further away from this initial introduction event, they became more genetically distinct. So, but the key here is that in both populations, we don't see any differentiation through time, which really suggests that that possibility of immigration was not true. Otherwise, we would expect there to be, individuals from different places, from different times in the same place clustering together. What about genetic diversity? Did the bottleneck actually reduce, did this introduction mean that Eastern house finches were less diverse and maybe more susceptible to infection? Well, we actually do see a decrease in genetic diversity, 4.2%, but when we compare the Western and the Eastern. So there was a reduction, not a lot, but there was something. But even more interesting is we actually see a 1 to 2% reduction in genetic diversity after the epizootic, which means that not only was this what we call the census population size, but actually genetic diversity was reduced due to this disease. Interestingly, 20 years after in the Eastern population, we see a lot of that genetic diversity coming back. And so that means that maybe there's some kind of local mixing, enough that where we might have had very local bottlenecks, over time, that reduction wouldn't really come into play. Why does this matter? Well, you might think 60% of individuals being lost is a lot, right? That would be very scary, especially from a conservation perspective. But actually, at the core of it, in terms of genetic diversity, it doesn't really have much an effect. So, just something to think about when we think about, how many individuals are being lost in different places. Okay, so back to our question. So I told you we could cross off immigration because we don't see any differentiation through time. So that just leaves polygenic selection, which again, just to remind you, that's change in frequency at alleles already present in the population at multiple sites. So these very subtle allele frequencies. Now, one thing that I didn't mention when we were looking at sweeps and how they differentiated through time it's not just this one site, so not just these Cs that sweep through time, but also any sites that are linked to them. So we can use a number of different statistics, so something more powerful than just looking at changes through time. I'm not going to go into the details of these, just know I'm happy to talk about them later if you're interested. But we can look at differences in a variety of different statistics to really have more power to look at how these these alleles might be differentiated. And so when we look at kind of Eastern before versus 10 years after, Eastern before 20 years after, Western before 10 years after, there are a number of different genes in the genome that pop up as being differentiated according to all of these summary statistics. And that shows that polygenic selection has occurred. One cool thing is that the genes we see in the Western population are different from those that we see in the Eastern population. So it looks like, these two populations are adapting to MG in a different way. So I've just told you a lot of information, a lot of science here. Looks like polygenic selection occurred. But just to remind you, I told you that pathogens are one of the most consistent selective pressures across birds. Viruses are shared selective pressures in birds and mammals. And selection in recent timescales in population results in long-term signatures of positive selection. And finally, that rapid adaptation occurs at many different places in the genome, termed polygenic selection. Now, I'm really excited to continue this into the future. You might think, why do we care about how house finches evolved resistance to Mycoplasma? But actually, the dynamics of how this disease has kind of swept through house finches and how house finches have responded is very similar to what we're seeing in terms of how COVID is sweeping through human populations and how human populations are responding. And so we might look at what's happened in house finches as maybe a window into the future in terms of what's going to happen with COVID. So, learning about these things in birds actually has broader implications elsewhere. I just put in a grant for a disease I have a future work that I'm working on now with Dr. Susan Ballinger and a few other researchers, where we're really going to try and integrate not just evolutionary history of the host, but also look at the evolutionary history of the pathogen, as well as host ecology, their community, life history, individual variation due to stress, doing environmental differentiations, microbial communities, so that we can see, really see, how has one species going to respond to diseases from that particular species, and also pathogens from more distantly related species, so that we might predict how future outbreaks and future epidemics might be changed in the future, what might happen in the future. So we might predict what's going to happen. So I just wanted to end with my acknowledgements. Thank you to everybody my collaborators on these different projects, museum collections, all those who helped me out in the field collecting samples in the lab, computation, museum. And I am more than happy to take any questions that you might have.
Well, thank you very much, Allison. We really, really appreciate it. And yes, we have a question from Kathy Ellsworth. Kathy asks, we've heard of the phrase herd immunity a lot recently. Where would herd immunity fit in your analysis?
Yeah, that's a great question, Kathy. So, one thing to think about with house finches is, just like us, how does the disease is spreading through social contact and through actually at bird feeders. And so, where are our house finches coming into contact with each other? It's actually at bird feeders. And so, basically what this herd immunity is the fact that the house finches stopped dying after 2 years. And so, we're basically accomplishing that not through humans dying, but some humans have died, which is very sad, but through immunization. And so that's, I've talked a lot from an evolutionary perspective. We're talking about years here. From kind of the vaccine immunological perspective, another kind of arm of the immune system is the adaptive immune system, which is going to basically be our immune system that's going to recognize something we've already been exposed to, and then going to it, and then our immune system's going to fight it off before it has a chance to take hold. And so, we're seeing that in house finches, they're not Mycoplasma is still there, but it's not killing them anymore, and it's not giving them as severe of symptoms. So basically, we could think of as enough house finches have been infected to A, they've got resistance now to the disease, and B, they're exposed and kind of there are enough of them that have this previous exposure that they're not carrying it to other house finches. So it's kind of like a two-pronged approach. So I hope that answers your question, at least. So it's a very complicated subject. Yes, flock immunity. I like that.
Flock immunity. Yeah.
Only birds could wear face masks. We can't tell them to socially distance from each other.
Yeah, they need goggles.
That's right.
Kathy kind of follows up with that. It seems that immigration is not the most likely to result in extinction. Is that true?
Well, I think she says immigration is the most likely to result in extinction.
Is the most likely. Yeah, I'm sorry.
Yeah, it really depends. So, it's a very interesting question from a conservation perspective. There's been, especially if there's a very limited population size. So say something like Florida scrub jays. This has actually been studied a lot. There are only a few hundred of those left. People worry a lot about like a disease coming through and just wiping out all of the Florida scrub jays. And so one thing that people have been thinking about is actually introducing individuals from outside. And what some researchers have found is that you actually have to worry about introducing too much genetic diversity into these small populations from outside, because they might be maladapted to where the small population is existing. So it's a very it's a very touchy subject, and it's not just more genetic diversity is always better. It really depends on where these individuals are coming from and kind of how precarious it is for that particular population or species.
Fantastic. We also had a comment from Lily. She says, "Hi, Allison." She's one of our lab student birders, and she had met you at the last Nature Fest.
I remember you, Lily.
And she's really glad you're here.
Yeah, it's nice to see you kind of in the chat room at least.
Okay, I'll let you. You can.
We have another question. Yes. Yeah. So, are there interactions where genes in an individual would improve individual fitness but be harmful to the population as a whole? So, for example, if they were less affected by MG symptoms but still passed it on rather than dying. Yeah, absolutely. And one thing to keep in mind is that selection in these genes to be more resistant to MG actually might have negative consequences for other stressors. And so this is, this big push and pull where I might be able to survive this disease better, but the same variance is going to make me more susceptible to a different disease, for example. And absolutely, that's just like, we can see in carrier individuals in COVID, as an individual, I might not be as sick, but I might be able to pass it along to others. And that's absolutely true in birds as well, in house finches or any other birds. If you have the disease and you don't have the symptoms, and so you're not changing your behavior at all, then that could definitely negatively affect the population.
Absolutely. Do we have anything else?
Yes, Bella has a question on the.
Oh, no, that's one we just answered.
Yep.
This is Frank. I have a question. I was really fascinated by the data you showed on the interrelationship between birds, mammals, and different viruses. And you pointed out coronavirus, but also the one of interest is influenza virus, and how there's this interplay between the virus and sort of evolutionary selective pressure on people and birds. And there's a very complex relationship. Can you say anything about that in terms of what that means for birds and for humans?
Yeah, so from the bird's perspective, so influenza A is an interesting one in that it actually does make some birds very sick. And so there are some variants of influenza that have mild consequences for birds and that have severe consequences, just like influenza for us. One possibility is that if influenza can go into a reservoir like birds and it can evolve, it can mutate to become very virulent in birds. And, they might have this kind of host pathogens arms race, and say that variant evolves to become very virulent. And there's, it can jump to us or, another mammal or another bird species, for example, that could have very devastating consequences, and vice versa. The same, we think of things from like a human-centric perspective a lot of times, but it's also possible that, something could evolve to become much more virulent in us and then jump to birds. And because there's this, it's not necessarily very easy for a disease like influenza to jump from one species to another, but it's much more common than it might be in other cases. And so, it's very complicated, but it just shows how important it is to look at these evolutionary studies and understand, how often has this exchange taken place, and then also to understand, how do the different variants from influenza impact species that are kind of have variable evolutionary relationships to each other. Did that answer your question, Frank?
Yes, I think, yeah, it did.
In some way.
It's a very complicated subject, but I think
It is.
it's an important one for future study for sure.
Absolutely. Yeah, I'm very excited I hope this grant gets funded with my collaborator, Susan, and I. Microplasma, we think of it as a house finch disease. In chickens and turkeys and poultry, it doesn't give them the conjunctivitis. It just gives them a respiratory infection. And so one thing that has been found is that it's actually probably much more common across songbirds than we know about, but just nobody's looked at it, right? And so how common is MG across birds as a whole, and how infectious is MG, say, from a chickadee to a house finch? And so, those types of questions can help illuminate questions of influenza A, where it might be very difficult to study because it's so infectious to humans. So we have a couple more questions. So James Zimmer says, birds mix genes sexually, disease by original mutation, one an advantage over the other. I'm not exactly sure the question here. So I think maybe what James is saying is how what's called recombination, which is how the genome kind of mixes every time you have a new organism being formed due to what we call crossing over, where you have two copies of your chromosome, and some of the chromosome stays over here and some of the chromosome stays over here, and there's interchange. And so what that recombination means is that it's going to basically break down how genes are linked together on a chromosome. And so if you have a mutation that's going to be spreading throughout the population, and you don't have any recombination, there's a possibility that the mutation, it might be linked to other genes that have other important functions. And so it might be this gene might be advantageous, but maybe these other genes it's linked to might cause problems. And so recombination is actually what can separate those through time, through when you have these chromosomes mixing together. Actually, dogs are a great example of this, because some of the genes for the traits that people select for, like wrinkly skin, for example, is linked to genes that cause health problems. So, certain diseases, or certain breeds are more likely to have certain health problems. And it's because the genes for the traits that are selected for in that breed are linked to other genes that are responsible for the disease. So yeah, it's a
good argument.
Subject.
Yes.
That's right. And so I see Kathy has a question. So referring again to that chart, it seems that coronavirus doesn't cross from mammals. Can you show that chart again, please? Absolutely. Let me just go back to it in my presentation real quick. Yeah, so there are different kinds of coronaviruses. like delta coronaviruses and gamma coronaviruses. And the coronaviruses that birds have are a completely different kind than the kind that mammals have. So I'm just going to show this here.
Oh, okay.
So here we've got, here are mammal coronaviruses. And you can see there's zero crossing over between birds and mammals. Here's the bird coronavirus. There is this one branch that's like mammals that's crossing over, but it's actually next to this fish one instead of a bird one, which is interesting. But another thing you'll notice is that almost all of the colors in here are blue and green instead of, and so that just means that almost all of what we've sequenced come from mammals and birds. There's probably a lot of other interesting stories out here in fish, reptiles, and amphibians, and vertebrates that just nobody's ever looked at it, and nobody's ever studied it.
Interesting, yeah.
And Kathy asks, what equipment do you use to determine the genomes? Well, I use specifically something called an Illumina sequencer. So basically, we extract DNA from a tissue, could be a blood sample, could be a tissue sample, and then you break it up into small pieces because our DNA sequencing technique that we use, it sequences actually very short fragments, actually about 150 to 300 base pairs. And then you have what's called a reference genome, where you've got these little fragments already assembled together in the order that they're supposed to be, and you can map them back to that reference genome. And that's going to give me the places where they differ between individuals, basically. So if you ever hear about genome sequencing or Illumina sequencing as applied to humans, I use the same exact technique, but just applied to birds. One fun trick is actually to use tools that are developed for all of the research done on humans and take advantage of those and put them into research on birds. So it's a way we move the field forward.
Fantastic.
You're a human microbiologist. You know probably what Illumina sequencing is.
I have a question about a virus. In this case, is West Nile. West Nile came in and just devastated across the corvid population in Southern California. It's also a human pathogen. So what's happened? It seems like the corvids have sort of rebounded from that. And does that have any implication for humans?
Yeah, so West Nile is actually a disease that I would very much like to study in the future. In past talks, I've talked about future research. My computer is giving me the beach ball of death, but if the slide actually advances, it's okay. It'll get there. Here we go. So West Nile is a very interesting one in that it was introduced to North America in 1999. So very similar to MG, but instead of just impacting one species, house finches, it impacts a whole bunch of different species. So here, this was research done by George et al. In 2015, and actually research out of Tom Smith's lab at UCLA, where you can see some species had kind of a very dramatic drop due to West Nile virus, but recovered very quickly, like the white-eyed vireo, the Carolina wren. Some species have had a decline and stayed declined. Some species, like who would have guessed the wren-tit actually declined due to West Nile virus? I wouldn't have guessed that. We hear a lot about corvids, but it's actually true across the bird family tree. So this will be a really cool one to study in terms of a number of different birds that have had persistent West Nile impacts and temporary West Nile impacts to understand, are these species resisting West Nile or evolving in the same way across the bird tree of life? Now, your question was how this impacts humans. Well, if birds evolve to where they can basically tolerate West Nile virus, and so it can just be circulating among different populations of birds, then that means that it might be much easier to have this reservoir that can jump then to humans later. And West Nile virus, the other possibility is that West Nile virus might evolve to become, it might evolve so that it can't jump to humans. It's who knows what's going to happen with evolution, but there's that possibility as well.
So do you
Interesting.
sequence the viruses or the mycoplasma as well as the bird genome?
You know, I haven't done that in my research, but it's what I'm looking to do in the future. But others in the Edwards lab have sequenced mycoplasma. I don't think many people have sequenced West Nile virus. I could be wrong. I haven't looked into this much yet, but there's some interesting work done in terms of how mycoplasma has actually evolved. It's when it jumped. So it looks like, for example, there was a single jump from poultry to house finches. If we look at the family tree of mycoplasmas, they just form a single group. So which means that it wasn't like multiple jumps of house finches being infected. It just occurred a single time. And then it's evolved very quickly. And, you know, one thing we've learned is that virulence is evolving to become greater through time. So when it first jumped to house finches, it had big impacts on house finches because they had no resistance and tolerance. But, you know, as they have evolved to become resistant, as well as they have this, you know, herd immunity due to individuals being exposed in their lifetime, the mycoplasma is having to evolve to become more virulent. So we may see, you know, repeated kind of cases of this outbreak that like we saw in 1994 when it first jumped to house finches over and over again. And we actually have seen in the West that the Western strain, actually the one that first jumped to house finches in the West, was not very virulent at all, but that's also evolved to become more virulent over time. So it's a really fascinating disease to study.
Interesting. Well, I think we've run out of questions. Allison, thank you very, very much for a wonderful, wonderful talk. I'm sure that many of us will be looking at the recording on our website.
Yes, I'm sorry it was less bird pictures than you're probably used to, but
no, actually, we appreciate your talk a lot. It meant it's the kind of talk that we exactly were looking for. Thank you very much.
Yeah,
well,
thank you very much.
Yeah, you'll have to bring me back to talk about bird color sometime. Then I'll show lots of beautiful pictures.
All right, that sounds great.
Take you up on that.
Absolutely. I just want to remind everyone that next week at this same time, same station, well, maybe a different station. We'll have a different link.
Same station.
Same station. We will have John Feenstra on kingbirds, part of our ID series. And next month, on the first Tuesday of the month, we'll have Cinti Lee, who will be talking about the Impidinax family. And it's a very timely topic. And I hope you can join us then. And once again, Allison, thank you very much. We appreciate it.
Thank you. It's been a pleasure.
And I love your background. You have to share that with us.
Thank you so much. It was very informative, really great.
Glad you enjoyed it.
Thank
Absolutely.
you, great.
All right.
Okay, with that, we'll see you all next week. Next week and next month. Thanks again. Take care, everyone.
Bye.
Bye-bye now.
Goodbye.